Welcome to the Fatty Acid Forum, sponsored by Virtus Nutrition. Let's listen as Dr. Charlie Staples from the University of Florida discusses how the essential fatty acids are stored in bovine tissues.
In our continuing webinar on essential fatty acids, today we're asking the question, how are the essential fatty acids stored in bovine tissue? Essential fatty acids are stored in animal tissue in either a triglyceride form or a phospholipid form. First, we'll consider the triglyceride storage form. As a reminder, triglycerides are composed of one glycerol backbone and a single long-chain fatty acid that's attached to each carbon of the glycerol, so that there are three fatty acids in a triglyceride molecule. In this example, the top fatty acid is palmitic. It's a saturated fatty acid. The second is oleic with one double bond located there in the middle. And the third fatty acid is alpha-linolenic acid with three double bonds. And triglycerides are the main storage form for many fatty acids in adipose, muscle, liver, milk, etc. And the body can mobilize these essential fatty acids in storage as they're needed. The second way that fatty acids are stored is in phospholipids. And phospholipids are composed of one glycerol backbone, two long-chain fatty acids linked to glycerol by ester bonds, and an organic compound such as choline, which is linked by a phosphate bridge. The structure of phospholipids is illustrated here. The pink box is glycerol with the three carbon molecules with fatty acids attached to two of the carbons, and at least one of those fatty acids is unsaturated. Phospholipids tend to be rich in omega-6 fatty acids. A polar group is attached to the third carbon molecule of a glycerol by a phosphate bridge, which is indicated here in orange. And the polar group can be choline, it can be ethanolamine, serine, and inositol, or sphingosine, as listed here across the top. A phospholipid called phosphatidylcholine, as shown on the left, has choline as the polar group. If serine were the polar group, then the phospholipid would be called phosphatidylserine. Now, both triglycerides and phospholipids are found in the same tissues. For example, in the longissimus muscle of lambs, the concentration of triglyceride was 2.4%, and that of phospholipids was 0.38%, or grams per 100 grams of meat. The long-chain fatty acid profile differs between the triglyceride and the phospholipid, and these pie graphs show the proportion of each fatty acid in triglycerides and phospholipids in this muscle using the color code here on the side. The dominant fatty acid was linoleic acid, followed by C16-0, which is palmitic acid, followed by stearic acid, C18-0 in red. And these three non-essential fatty acids made up about 94.5% of the fatty acids in triglycerides, but they only made up 61% of the fatty acids in the phospholipids of the muscle. On the right side, the concentrations of the essential fatty acid C18-2, linoleic acid, in purple, was substantial in phospholipids, being 16.1%, compared with only 2.1% of the fatty acids in the triglyceride form. The concentrations of the essential fatty acid C18-3, linolenic acid, in light blue, was 3.4% in the phospholipid fraction, but only 1.1% in the triglyceride fraction. And only phospholipids had significant concentrations of the C20 and C22 fatty acids, as indicated by the light blue and the pink color, 4.5 and 1.8% respectively in the phospholipids, and much lower concentrations in the triglycerides. Now, not only do the fatty acid profiles differ between the triglycerides and phospholipid fractions, but the concentration of the various types of phospholipids vary among tissues and milk, as illustrated in this table. The type of phospholipid is listed in the first column, and the type of tissue appears in columns across the top of the table. In bovine liver, the phosphatidylcholine was the major phospholipid at 52.5%, and second was phosphatidylethanolamine at 30.5%. These same two phospholipids were most dominant in the heart tissue of unweaned calves as well, about 45 and 29% respectively. These two phospholipids were found in similar dominant concentrations in the uterine endometrium of ewes and the intestinal brush border membrane of rats. In bovine milk, phosphatidylethanolamine was the dominant phospholipid with significant amount of sphingomyelin. So the point is that the proportion of the phospholipids are different across the different tissues in milk found in animals. Now, in addition, each phospholipid has a different fatty acid profile as shown in these pie charts of the phospholipids, in this case in the uterine endometrium of the ewe. The saturated fatty acid in blue and red combined, C16 and C18-0, only make up about a third of the fatty acids in the most common phospholipids. Phosphatidylcholine make up about 37%, and phosphatidylethanolamine makes up about 32%. So most of the fatty acids are unsaturated in phosphatidylcholine and phosphatidylethanolamine. Similarly, saturated fatty acids only made up about 45% of the fatty acids in phosphatidylserine. The exception is phosphatidylinositol, for which 2/3 of the fatty acids are saturated, the 8 and the 58.9, blue and red there for phosphatidylinositol. Now, linoleic acid, C18-2, the purple slice, is as high as 12.4% in phosphatidylcholine and as low as 2.5% in phosphatidylinositol. Concentrations of linolenic acid, C18-3, which is the turquoise color, is much lower than linoleic acid. It is as high as 0.4% there in phosphatidylcholine and as low as 0.05% in phosphatidylserine and inositol. Only phosphatidylethanolamine has significant amounts of the longer chain omega-3, C25 in light blue and C22-6, which is DHA in pink. You'll notice that concentrations of C18-0 stearic, the red, increases as concentration of C18-2, the purple, decreases. So they're kind of inverse related in concentration. Now, phospholipids are the fundamental building blocks of all cell membranes. Because the fatty acid of the phospholipids avoid water, that is, they're hydrophobic, and the phosphate end of the phospholipids love water, they're hydrophilic, they can spontaneously form a bilayer, or in other words, two layers of phospholipids. So in this diagram on the left, the green bars are the fatty acids, and so those fatty acids come together and they are hydrophilic, and then the phosphate groups are on the outside. It's indicated by these spheres, and those spheres or the phosphate ends are hydrophilic, they love water. So the fatty acids stay together, and this forces the phosphate ends to be toward the outside. Well, this cell membrane is very thin. If an animal cell was enlarged to the size of a watermelon, the membrane would be the thickness of a piece of office paper. This characteristic of attraction to or aversion of water spontaneously causes this sphere to form like a cell. And this is somewhat similar to the spontaneous formation of soap bubbles. In that case, the hydrophobic parts face outward and soap bubbles instead of inward. But in this arrangement, water is trapped inside the cell, and there's almost no water within this bilayer. And this water-free environment of the cell membrane prevents most water-soluble molecules like sugars or salts or ions from entering the cell on a willy-nilly basis. Rather, those movements of those water-soluble compounds are controlled by transporting mechanisms. Now, these phospholipid molecules within a monolayer of an animal cell membrane can rotate and move laterally. It's illustrated here with the red polar group and the two fatty acids shown moving to the right and to the back. In addition, the blue phospholipid shows that it can rotate. And these fatty acids can change places with their neighbor very rapidly, about 10 million times per second. Now, because the animal cell membranes contain phospholipids of mainly unsaturated fatty acids, the membranes are fluid, that is, they're soft and flexible, unlike plant cell walls that are rigid due to the presence of fiber. The double bonds in the fatty acids introduce kinks in the fatty acid chains, making the fatty acid more difficult to pack together tightly, and this allows the cell to move more freely. So as an example, say compare butter versus vegetable oils. Vegetable oils contain more unsaturated fatty acids, so they are liquid at room temperature, whereas butter contains more saturated fatty acids that pack tightly, and so it is solid at room temperature. Now the phospholipids make up about 50 to 60% of the cell wall, with the rest being mainly proteins and cholesterol. So the blue structures in this diagram are types of proteins, and the yellow represent the cholesterol. So the blue structures in this diagram, as I said, are the protein structures, and they're necessary for proper cell function. Why do membranes contain such a wide variety of phospholipids and with polar groups that differ in size and shape and charge? What role do these different phospholipids play? Well, we're just beginning to learn more and more about those. We do know that we can change the fluidity of the cell by changing the fatty acid profiles. By changing the fatty acid profile and changing the fluidity, we do affect the binding of some proteins in the cell wall. We affect the fusion of some proteins and other biomolecules within the membrane. Therefore, we affect the function of these molecules. Certain protein bodies within the animal cell membrane only function in the presence of certain phospholipids. The presence of other phospholipids can indicate the health of the cell. For example, the migration of phosphatidylserine from the inner to the outer part of a bilayer of a cell membrane helps identify that cell as dead, and this signals macrophages in the animal's system to engulf and digest the dead cell. So in summary, essential fatty acids are common components of all cell membranes in the form of phospholipids. The tissues contain all or most of the many phospholipids, like choline and inositol, but in different proportions with different fatty acids. The phospholipids contain our essential fatty acids, and these essential fatty acids, they improve fluidity of the cell membranes, they improve the functioning of the cell surface proteins that are present, and they also influence the ability of cells to speak with or influence one another.